Natural killer cell functions. Natural killer cells are the ultimate security guards of your immune system. First, recognition. Natural killer cells are on a mission to target and kill virus-infected and tumor cells. But how do they avoid attacking normal tissue? Through inhibitory signals. By binding to the MHC1 receptor on healthy cells, they get the green light to stand down and spare them. The destruction mechanism by perforin and granzyme. When they do spot a target, like a cancer cell, they release powerful granules. First comes perforin, which literally punches holes, or pores, into the target cell's membrane. Then, the granzyme pathway kicks in, activating caspases to force the enemy cell into programmed cell death, or apoptosis. Next is ADCC and cytokines. Natural killer cells also exhibit ADCC function, or antibody-dependent cellular cytotoxicity. They use their CD16 receptors to recognize and destroy cells coated in antibodies. Finally, they are not alone. They use cytokine secretion, releasing interferon gamma to recruit and activate macrophages for backup. Natural killer cells execute two of their most powerful mechanisms for cell destruction, ADCC and cytokine production. ADCC function. ADCC, antibody-dependent cell-mediated cytotoxicity. Activated natural killer cells, which express CD56 and CD16, use that CD16 receptor to lock onto IgG-coated target cells. This binding interaction triggers immediate degranulation. Second mechanism of killing. Once triggered, the natural killer cell releases perforin and granzymes. Perforin acts like a drill, punching holes right through the target membrane, allowing granzymes to slip inside, activate caspases, and induce apoptosis to destroy the target. Third mechanism, cytokine production and cooperation. But natural killer cells don't stop at direct killing. They are major players in immune system cooperation. They produce high levels of interferon gamma, which potently activates macrophages, upregulating phagocytosis and microbial killing. They also release other cytokines like tumor necrosis factor, alpha. By cooperating with T cells, they seamlessly regulate the adaptive immune response. Regulation of natural killer cell cytotoxicity. Natural killer cell activation is tightly regulated by a sophisticated balance of opposing signals derived from clonally distributed inhibitory and activating surface receptors, which ultimately determine the threshold for cytotoxic degranulation. Scenario 1. Immune tolerance and the inhibitory signal. Consider Scenario 1, illustrating immune tolerance toward a healthy autologous host cell. Natural killer cells constitutively express inhibitory receptors, predominantly killer cell immunoglobulin-like receptors, or KIRs. Concurrently, normal nucleated host cells express robust levels of major histocompatibility complex class I molecules. Mechanism of tolerance. Engagement of MHC class I by inhibitory KIRs delivers a dominant negative intracellular signal, effectively breaking the natural killer cell and preventing autologous lysis. This paradigm forms the basis of the missing self hypothesis. In second scenario, pathological alterations and activating pathways. Conversely, Scenario 2 demonstrates the response to malignant transformation or intracellular viral infection. Pathological cells frequently down-regulate surface MHC class I expression to evade CD8+ T cell surveillance. Simultaneously, cellular stress induces the upregulation of specific activating ligands. Activation and effector function. The concomitant loss of the dominant inhibitory signal, coupled with the engagement of activating receptors by stress ligands, shifts the cellular equilibrium toward full activation. This signaling cascade triggers immediate degranulation, releasing perforin and granzymes to induce targeted apoptosis of the aberrant cell. Natural killer cells function in continuous immunological surveillance, recognizing altered self phenotypes. Primary target identification relies on the absence of surface MHC class I molecules or the expression of cellular stress ligands. First, Natural killer cells eliminate virus-infected cells that downregulate MHC class I to evade adaptive immunity. This action occurs during the early, innate phase of infection and is critical for controlling herpes viruses, such as Epstein-Barr virus and cytomegalovirus. Second, they participate in tumor immunosurveillance, targeting cancer cells displaying genetic mutations or tumor antigens prior to adaptive immune maturation. Finally, natural killer cells mediate the rejection of allogeneic grafts via the recognition of mismatched MHC class I complexes. Natural killer cell activation. Upon stimulation by cellular stress signals or antibody-coated targets via ADCC, the CD56+ natural killer cell initiates intracellular cascades resulting in targeted exocytosis and degranulation. Secreted perforin monomers polymerize within the lipid bilayer of the target cell membrane, forming transmembrane pores. These aqueous channels facilitate the passive influx of granzyme proteases into the target cytoplasm. Intracellular granzyme B drives the downstream activation of executioner caspases and induces mitochondrial cytochrome C release, initiating programmed cell death. This apoptotic pathway maintains membrane integrity to limit inflammatory propagation and viral dissemination, serving as a critical axis in MHC class I independent tumor immunosurveillance. Natural killer cell activation is driven by stress ligands engaging activating receptors, initiating a phosphorylation cascade via SRC, ZAP70, and PI3K. This pathway is counterbalanced by inhibitory receptors carrying ITIM motifs. Once net activation is achieved, the natural killer cell mobilizes preformed cytolytic granules containing perforin and granzymes, executing regulated exocytosis. Following entry into the target cell, granzyme B proteolytically cleaves and activates caspases 3 and caspases 7. Concurrently, granzymes induce the mitochondrial release of cytochrome C, activating caspases 9 to culminate in controlled cell apoptosis. This functional output is upregulated by interleukins 12, 15, and 18, and suppressed by TGF-beta. Therapeutic applications. Given these precise cytolytic properties, natural killer cells represent a major area of exploration in oncology, specifically through the engineering of CAR-NK cells and therapeutic monoclonal antibodies designed to augment targeted tumor destruction. Natural killer cells versus cytotoxic T lymphocytes. Phenotypic differences and target recognition. Natural killer cells are large granular lymphocytes phenotypically defined as CD56+ and CD16+. Whereas cytotoxic T lymphocytes are small to medium CD3+ and CD8+ cells. Functionally, natural killer cells respond to the missing self phenotype via downregulated MHC class I and cellular stress ligands. Conversely, cytotoxic T lymphocytes require non-self antigen presentation on MHC class I molecules via a specific T cell receptor. Primary targets and killing mechanisms. While both cell populations execute perforin and granzyme-mediated degranulation to eliminate tumor and virus-infected cells, cytotoxic T lymphocytes additionally target wider intracellular pathogens and can induce apoptosis via the Fas ligand pathway. Natural killer cells lack CD3 and T cell receptors, utilizing a balance of inhibitory KIRs and activating NCRs without forming immunologic memory. Cytotoxic T lymphocytes utilize highly specific T cell receptors and co-stimulatory signals, ultimately generating long-lived memory cells for adaptive immunity. Natural killer cells serve as vital instruments in oncological immunotherapy due to their innate cytolytic activity against malignant cells displaying absent or down-regulated MHC class I. To enhance targeting precision, natural killer cells can be engineered with custom chimeric antigen receptors, or CARs, combining innate cytolytic efficiency with antigen-directed specificity. Therapeutic strategies also utilize antibody-dependent cell-mediated cytotoxicity, or ADCC, where monoclonal IgG antibodies like rituximab or trastuzumab coat malignant targets to stimulate CD16-mediated degranulation. Furthermore, allogeneic donor natural killer cell infusions provide an off-the-shelf modality to treat refractory hematologic malignancies, such as AML, while minimizing graft versus host disease risk. Current research focus is directed toward overcoming immunosuppressive factors within the tumor microenvironment, such as tissue growth factor beta and checkpoint ligands, optimizing cytokine production, and reversing natural killer cell exhaustion to sustain long-term clinical efficacy.